Dear students, in previous lectures we have generally discussed about sterilization techniques. In today's lecture we will more precisely discuss about plant tissue culture sterilization. So, as you all know that in plant tissue culture that involves small pieces of plant tissues which are known as explants, and they are cultured on appropriate nutrient media under appropriate growth conditions. Appropriate nutrient media that is good for producing callus, somatic embryos, suspension cultures, and new shoot and root. So there is a hormonal balance that various nutrient combinations of nutrients, minerals, plant growth substances, vitamins, and sugars. These are basically suitable for the biological contaminations, and they allow them for the rapid growth of bacteria and fungi. So if there is rapid growth of bacteria and fungi, that results in depletion of nutrients for the growth of explant, and there is also production of various toxins. So there is a need to sterilize your culture media and culture containers. There is also need to sterilize the seeds or plant tissues which you need to be cultured, and there is also a need to sterilize the instruments which are used to handle and ventilate the plant tissues, and also the rooms or cabinets. Sterilize the rooms or cabinets and filter air environment like laminar airflow. So, killing or removing all kinds of microorganisms in materials or in objects is known as the process of sterilization. Aseptic sterilization techniques, media, and apparatus are rendered sterile by autoclaving at 121 degrees centigrade for 15 minutes. Sterile plasticwares. Filter sterilization is employed for heat-level substances, while aseptic transfer by using standard bacteriological techniques using laminar flow hood is conducted. So first of all, we will go for the sterilization of culture rooms and transfer hoods. Tissue culturing is basically, there is a first utmost requirement is the compartmentalization of the lab work. So there should be compartmentalization into work elements. There you have to specify that which area is specific for the media preparations, glassware washing, sterilization, microscopy, and aseptic transfer. Large transfer rooms you can easily sterilize using the UV lights, and there is also need to wash them once or twice within a month with commercial brands of antifungal liquids. Small transfer rooms or hoods can be sterilized using UV light, and laminar flow hoods are usually sterilized using 95% ethyl alcohol and UV treatment for 15 to 20 minutes. Culture rooms should be initially cleaned with detergents and then carefully wiped with 2% sodium hypochlorite solution or 95% ethanol. Formaldehyde gas is excellent disinfectant for large culture rooms, or formalin inactivates the viruses and bacteria by inactivating the proteins. So fumigation is another best option for large area laboratories. Most commonly methods that are used to decontaminate the tissue culture rooms. If we talk about the sterilization of nutrient media, then there are two different kinds of sterilization. First is the steam sterilization, then second is the filter sterilization. In case of steam sterilization, nutrient media are autoclaved and at 121 degrees centigrade for 30 to 40 minutes. However, there are some problems associated with this sterilization, that if you sterilize the media in autoclave, then there could be following changes that could lower your pH, and that could also caramelize the sugar, which may have toxic effect, and there may be some volatile substances and salts may be precipitated out. And in that case, maybe salt might precipitate down. In case of filtration, Sterilization, there are the proteins, vitamins, and amino acids which are heat labile that can be sterilized using filter sterilizations. Millipores, nalogens, or seeds filters are basically used with porosity no longer larger than 0.2 microns. Most filters are of cellulose acetate or cellulose nitrate and available pre-sterilized plastic decomposable units. So nutrient media with thermolabile components should be sterilized using the filter sterilization. After this, the solution of heat-stable components is sterilized by autoclaving and cooled to 50 to 60 degree. Then thermolabile components are filter sterilized, and then these solutions are combined under aseptic conditions to give the complete media. So here is the flow chart for the explant of tissue culture. A few recent advances have also been introduced, such as plasma sterilization, in which low temperature hydrogen peroxide gas plasma sterilization is performed. Ionized gas applications of electric field to gas results in ionization into electrons and ions that utilizes low temperature, low pressure environment to create a unique plasma state that eradicates the microorganisms through combination of physical and chemical processes. Another is the pulse light sterilization. Here is the first patent that was published in 1984, and pulse is produced in 2 steps. A 20 kilovolt pulse lasting a few nanoseconds makes the xenon filled lamp, and then the capacitor charged at 3000 voltage discharge and make this off during the 3300 microseconds pulse, which ionizes the gas lamp and generates a pulse emitting a very high intensity white light. Another is the hydroclave, is also used for the sterilization of instruments. For the procedure for cleaning the glasswares, first of all you have to soak the glass in 10% soap water for 1 hour, then transfer the glasswares into a concentrated HCl for 2 hours. Then you will rinse water in glass in tap water, and then for 2 hours you have to keep glassware in 140 to 160 degrees centigrade. The surface sterilization of explant is very much